Hello students, in today's class we are going to learn about stages of prokaryotic transcription process. So basically transcription in prokaryotes can be divided into three main stages: initiation, elongation and termination. Now coming to the initiation stage, it is divided into two substages: binding and actual initiation. During the binding phase, there is initial interaction of RNA polymerase with the promoter region that leads to formation of a closed complex. Then a 12 to 15 base pair region of DNA from within the -10 position to +2 or 3 position is unwound, leading to formation of an open complex. We'll look at this in more details with the help of a diagram. So here we can see that initially we first have the complex of RNA polymerase holoenzyme, which consists of the core enzyme that is made up of the 5 subunits and the sigma factor, which is the 6th subunit. Together they form the RNA polymerase holoenzyme. So the sigma subunit here is going to help the RNA polymerase core enzyme to identify the promoter regions and to decide on which sequences are to be transcribed by the RNA polymerase core enzyme. So the process begins by binding of RNA polymerase holoenzyme near the promoter regions. So initially when the DNA binding when the binding of RNA polymerase holoenzyme to the promoter region takes place, the DNA is still in a double-stranded form and hence it is known as a closed complex. Later on, as the RNA polymerase binds more tightly to the DNA, there is unwinding of DNA leading to opening of the double-stranded DNA or formation of a bubble. As a result, the complex here is known as an open complex. Once the open complex is formed, then the RNA polymerase the RNA polymerase core enzyme can begin with the initiation stage, wherein it starts adding or polymerizing ribonucleotides, forming a short stretch of RNA. As soon as a short stretch of RNA of about 50 to 70 ribonucleotides is synthesized, then sigma subunit leaves the RNA polymerase uh, core enzyme to continue with the elongation steps. So now the elongation stage will continue. So elongation in case of prokaryotic transcription is nothing but further polymerization and addition of bases till uh, they reach at specific sequences where the transcription process is to be terminated. In case of prokaryotes, two different types of transcription termination signals are recognized. One class relies on a protein factor which is known as Rho, and the other class is Rho independent. So Rho independent transcription terminator, they basically have two differentiating features or distinguishing features. Firstly, there is a region that produces an RNA transcript that is self-complementary in nature, leading to formation of a hairpin-like structure, which is present about 15 to 20 nucleotides before the projected end of the RNA strand. So we'll understand this more better in later slides. The second feature is also presence of a highly conserved string of adenine residues in the template DNA that is transcribed into uracil residues in the RNA. So RNA polymerase basically takes a pause or halt at when there is formation of the hairpin loop-like structure, uh, when it is immediately followed by a sequence of adenines in the DNA and corresponding uracils in the RNA, that leads to transcription termination. So we'll look at it again with the help of this diagram. So this is your template strand that runs in 5' to 3' direction, and the RNA that is synthesized in 5' to 3' direction. So your RNA polymerase will be present here, and this is the RNA that is synthesized. So where the transcription termination is to be taken place, two types of sequences are important. First is the hairpin loop-like structure. As you can see, towards the 3' end of the messenger RNA or any type of RNA, you can see that there are some sequences that are self-complementary in nature. So they can base pair with each other, forming a stem-like structure, while there are the other sequences that are not complementary to each other. So they cannot base pair with each other, leading to formation of a loop-like structure. Hence, the structure is known as a hairpin loop-like structure or a stem loop-like structure. Now, formation of this hairpin loop-like structure creates a kink or a disturbance in the RNA-DNA hybrid. However, the transcript cannot release directly unless there is a sequence of adenine residues that are present immediately adjacent to the hairpin loop-like structure. So the sequence of adenine residues in the DNA are transcribed into sequence of uracil residues in the RNA, which is a continuous stretch of RNA-DNA hybrid that is showing presence of double bonds. So the double bonds are also comparatively quite weak uh, against the triple bonds in guanine and cytosine residues. So a combination of these two types of structures, firstly, formation of a stem loop-like structure that decreases the stability between RNA and DNA hybrid, which is immediately followed by a stretch of double bonded sequences, further decreases the stability between DNA RNA hybrid, and as a result, the RNA simply falls off or loses its association with the DNA. Once the RNA has fallen off, the RNA polymerase cannot continue, and this leads to transcription termination. Since no other protein factor is required here, hence it is also known as row-independent transcription termination. The second type of transcription termination mechanism is known as row-dependent transcription termination because it requires a protein factor which is known as a row factor. So, row-dependent transcription terminators, they lack the sequence of adenines in the template DNA strand. Uh, on the contrary, they contain a CA-rich sequence, which is known as the root sequence or the row utilization elements. So the row factor basically associates with the RNA at the specific binding sites, and they migrate in 5' to 3' direction till they reach the transcription complex that is paused at the terminator site. So here it contributes to the release of RNA transcript by its ATP dependent RNA DNA helicase activity that promotes translocation of the protein along the RNA. So we'll again look at it with the help of a diagram. So firstly, you have formation of hairpin-like structure. Sorry. So first, the transcription will allow formation of RNA in 5' to 3' direction. So one of the important factors are the or the sequences are the row utilization sequences. So row factors will basically polymerize around these sequences. Six subunits of the row factor will polymerize, and it is basically an hexamer. So once it has polymerized, it can make use of ATP dependent helicase activity. So in this case, by formation of a hairpin loop-like structure, there is again a kink in the structure where RNA polymerase takes a pause. However, the transcript cannot be released by itself. So here the rho factor is involved, which makes use of the RNA DNA helicase activity, which is ATP dependent, and it melts the interaction between the DNA RNA hybrid. As a result, it allows the messenger RNA or any transcript to leave its association with DNA. Again, RNA polymerase then cannot proceed any further, and the transcription is terminated in case of row dependent termination. So that's it about prokaryotic transcription process. Kindly like and share if you like the videos, and subscribe to get direct updates on the uploaded links. Thank you.